Now, leading the presentation this evening is Dr. Brooke Huuskes and Emily Major. So before we start, a little bit more about our speakers. Dr. Brooke is a senior lecturer at Trobe University in Melbourne, and the aim of her research is to investigate the contributions of the immune system to the progression of kidney disease and create novel therapies that target the immune system to regenerate the kidney. And Emily is a researcher and student ambassador, completing her PhD under Dr. Brooke's supervision. And fun note, in 2023, Emily was actually the recipient of the Pamela Ratcliff PKD PhD scholarship grant, which was co-funded by PKD Australia and Australian Rotary Health. So for the past 2 years, Brooke and Emily have been trying to understand how the immune system contributes to kidney cyst growth, as well as high blood pressure in PKD. And excitingly, they have found that a specific part of the immune system is turned on during PKD. So tonight we'll be hearing a lot more about that and their research. Tonight's webinar is titled, "Can we target inflammation to slow PKD progression?" Dr. Brooke and Emily, over to you.
Thanks so much, Jess. That was a lovely, lovely introduction, and everyone can just call me Brooke. It's okay. No need for the doctor. So thanks again for the opportunity to present what we've been working on. Yep, you'll see we've been working really hard, so it's lovely to be able to share this with you all. So I too would like to acknowledge the country that Emily and I are on. Actually, Trobe University is on many countries, and we proudly acknowledge the traditional custodians of the land in which our campuses are located. And this specific research was conducted on the lands of the Wurundjeri people of the Kulin Nation. Over to you, Emily.
Okay, so we all know the kidney is that little bean-shaped organ that makes urine, but why exactly is the kidney so important? Well, the kidney actually does much more than just make urine. It acts as a filtration station and removes all the gross stuff from our bodies. It does this through the little filtration systems called nephrons, as you can kind of see the squiggly thing on the screen there. So to get the job done, the kidney actually has around 1 million of these systems working all at once. But where does the waste actually come from? Well, the kidney actually removes the waste from our blood. So every time the heart pumps, around 20% of that blood goes directly to the kidney. Now, this is almost the same amount as our brain gets, which just kind of goes to show how important our kidney actually is. So the kidney can filter around 1 liter of blood per minute, and over a day, it can make 70 to 80 liters of urine with all that waste, which our body reabsorbs most of the water. So we only urinate out the bad parts. But the most important role of the kidney is that it can also control our blood pressure. So in polycystic kidney disease, or PKD, there is a part in the DNA that becomes faulty, which results in the kidney changing. So these changes can result in large kidneys that don't work as well, fluid-filled cysts forming on the kidneys, as well as high blood pressure. So we've all probably heard the term high blood pressure, or have had a doctor speak to us, either being at risk or being medicated for it. But what exactly is high blood pressure? So high blood pressure is also known as hypertension, and as some of you may have heard before. So blood pressure is basically the pressure of your blood against your arteries. So have you guys ever used a garden hose and then put your thumb over the end? Do you remember the water coming out a lot faster, a lot harder, or the water went further? So that is what we call pressure. And the same thing is happening inside our bodies when our arteries become restricted and when we develop high blood pressure. Now, blood pressure reads in a form of 2 numbers, which you may have seen when you visit the doctors. There is a top number, which is called systolic, and the bottom number, which is called diastolic. So your systolic pressure is typically the larger number. This is the pressure of your blood in your arteries when your heart pumps, whereas your diastolic is typically a lower number, and this pressure is when your heart relaxes. Now, your blood pressure can actually fluctuate. So when you're exercising, your blood pressure may increase versus when you're relaxing, your blood pressure may decrease. And this is just to help the blood move around your body. However, as I mentioned before, PKD is known to have high blood pressure. But what is actually causing this high? Sorry, I forgot to mention something. So normal blood pressure is around 120 to 180, whereas high blood pressure is when you are constantly reading over 140 to 90. So PKD, sorry, moving on, I will talk about how high blood pressure and the kidney interact. So inside your kidney, there's this special system called RAS. So your RAS system actually controls your blood pressure. So in a healthy kidney, this system is so sensitive that if there is a little change, your RAS system can kind of affect that. So if they notice your blood pressure drops slightly, your RAS system activates, increasing your blood pressure. And then if your blood pressure goes too high, your RAS system counterbalances that and lowers your blood pressure. Now, in a PKD kidney, all the healthy tissues, well, most of the healthy kidney tissue is destroyed, which means this RAS system doesn't seem to work as well. This can then cause your blood pressure to maintain higher. So is this the only thing contributing to high blood pressure in PKD? Well, what's been found in the science field is that inflammation may have a key role in blood pressure. And what is being found is that people with chronic inflammation, so inflammation over a long period of time, are more likely to have high blood pressure.
Amazing. Thanks for giving us an overview of high blood pressure, Emily. I'm going to talk about what a cyst is. So this here is an ultrasound image that Emily has taken, and all of a kidney, and all of these black spots, they're all cysts on the kidney. But how will cysts form? How do they get there in the first place? And there's actually a lot of research going on to understand these processes. So I'll try and go through some of it now. But first of all, let's zoom out and look at the kidney. This is, as Emily said, a bean-shaped organ. Not really interested in all of that. What I'm really interested in is zooming up on it and having a look at this, the nephron. So blood comes into this little part of the nephron here called a glomerulus, and all the filter, kind of all the waste product and water comes out and goes through this series of connecting tubules. And ultimately, the tubules is where that water reabsorption happens. So by the time we get to the end of the nephron, we're only really kind of peeing out the stuff that our body doesn't need. But let's take a look at a cross-section of kind of one of these tubules. So you can see the cells are arranged in a circle shape, and all the urine will be flowing through the middle. And you can see that each of these cells has this little projection coming off it. So let's have a look in more detail at what this projection is. We call this a primary kidney cilium. So the primary kidney cilium, if we take a closer look at it, is basically a projection of the cell with lots of proteins around here. And that primary kidney cilium are really important for detecting what is going through those kidney tubules, and it signals back down to the cell. So it's like a little arm projection that signals to tell the cell what's going on in the whole kidney. And I think it's really, really cool that we know that the primary kidney cilium looks like this, because we've been able to use really, really cool and advanced imaging techniques to actually see this in the kidney. So here is an image. You'll see images like this later on in our talk, but this is a stain that we've done, well, not me, but a stain that's been done on the kidney. And you can see this white arrow is pointing to this primary kidney cilium. I think this is so cool that we can actually see what all of this is. Now, using more advanced imaging techniques, you can see two cilium here on two different cells of the kidney. And if we look really closely, now we're talking, you know, microscopes that are in the millions of dollars worth. We can get really close and have a look at what this cilium actually looks like. And so you can see this here is a cell of the kidney, and projecting from it is this primary kidney cilia. And you can see this is the kidney cilium has been cut in half. We're looking down the kidney cilium, and you can see that this is what it looks like, which is exactly what our little drawing looks like. So imaging can tell us a lot about the primary kidney cilium. And you might be thinking, Brooke, why are you talking about the primary kidney cilium? And it's because it's actually dysfunction. This kidney cilium is dysfunctional in polycystic kidney disease. So when the primary cilium is dysfunctional, we see an increased number of the cells within the kidney. And so this increased number of cells is what causes the cyst to grow on the kidney. So in autosomal dominant PKD, you can see that these kind of bulges out of the nephron, they are the fluid-filled cysts. And if we compare that to autosomal recessive PKD, you can see that the cysts are really just isolated to one part of the nephron. And in fact, autosomal dominant and autosomal recessive PKD are just two forms of polycystic kidney disease that are caused by genetic mutations. And there are lots of other genetic mutations that can cause polycystic kidney disease. So we know that the primary cilium is dysfunctional and that causes cysts, but how does it actually cause cysts, and how do those cysts get larger and larger as time goes on? Well, it's actually really complex, and like I said, there's a lot of research going on at the moment to understand this, because the thought is if we understand how cysts grow, we might be able to target these kind of pathways with drugs to stop cyst growing. But what we know so far is when a cyst grows, we see increase in the cell number, and we also get some cells dying. We see an increase in inflammation, so activation of our immune system. We see an increase in blood vessel growth, so these cells are proliferating and growing, and in order for them to do that, they need oxygen and nutrients, and so they get that from these new blood vessels that grow towards the cysts. And we also see an increase in kidney scarring. And these little blue purple cells around here, these are a cell type of the immune system, which we're going to talk about a lot more, called macrophages. And macrophages seem to be really important in this process. There have been experiments where macrophages have been taken out of an animal, and the animal has had polycystic kidney disease, and the animal that doesn't have macrophages doesn't have as big a cyst or as number of cysts. So something's telling us that, you know, the macrophages are really important in cyst growth. So Emily spoke about high blood pressure, and I've just spoken about cyst growth, and there's something that kind of links the two together. Not too sure if you picked up on it, but it is the immune system. So the immune system plays a role in high blood pressure, and it definitely plays a role in cyst growth. So what is the immune system? Well, it's your body's defense system, basically, because your body is constantly under attack. Every time we breathe, every time we touch something, something's trying to get into our body and kind of do damage. So our immune system has really evolved to be really specific and really, really good at detecting, deflecting, and destroying anything that kind of comes in and tries to attack us. And I've also added repair here, because what we want is if we get an infection or we cut our skin or if something happens, we want to be able to repair and regenerate that kind of tissue that's been infected. But our bodies as humans are particularly bad at repairing. And so I like this graph because it shows how kind of really in the evolution of humans and of species, you can see that things like sponges, you know, coral underground and under the sea, they're great at regenerating. Really good. In fact, if you cut a piece of sponge off and put it on the sand next to this one, a brand new sponge is going to grow. But they have terrible immune systems, so they're very susceptible to things like infection. If we go a bit further along and we're in this kind of fish reptile stage, they're still really good at regenerating. This little axolotl here, or Mexican walking fish, and this zebrafish here have amazing regenerative capacities. You can cut out some of their brain, some of their heart, some of their kidney, and they'll just grow it back. So they're very, very good at regenerating. And they have a little bit of an immune system, but we've sacrificed the ability to regenerate for a beautifully complex immune system. So we really have two types of immune systems that you might have heard of. One is innate and one is adaptive. So our innate immune cells, which you can see here, they're kind of roaming around in our blood and our organs all the time, just kind of checking things out to see if we have any nasties like a bacteria or a virus. And when we do have a bacteria or a virus, they're our first responders. They get to the site quickly of infection, they cause inflammation, and then usually they call for backups. And the backup is our adaptive immune system. They come in and make sure the job is done, basically. The bacteria, the virus is gone because of these adaptive immune cells. Also, our adaptive immune cells are the ones that remember an infection. So, for example, if we get a vaccine, it will activate our adaptive immune cells so that they remember what our vaccine was, so that when we get an infection again, they remember they've seen the infection before and they just can clear that infection really quickly. So it's a really beautiful system. But as I alluded to, Emily and I are really interested in macrophages because we're pretty sure they play a key role in the progression of polycystic kidney disease. So inflammation is really important in our wound healing response. And many things can cause it. Like I've mentioned, bacteria, viruses, fungus, a whole lot of range of things that shouldn't be in us, that get in us, will activate inflammation. And so an example is if we cut our finger, some bacteria might enter our blood. That activates the immune system and causes inflammation. But eventually that will all lead to healing, which is what we want. But does the body always heal in that nice little way? Maybe by my voice you can tell that I'm going to tell you it doesn't. So in kidney disease, most of the time there is no nasty. There's no bacteria, there's no virus. Sometimes it's a gene in the example of PKD that has gone on, but then we still get all of this inflammation. So how does that happen? Well, when a kidney is injured, and it can be injured through lack of oxygen, through toxins, through stresses like high blood pressure, the kidney cells will die and they send out help me signals. And what that does is that causes a lot of cells of the immune system to go towards the kidney. Now, what happens is the kidney gets overwhelmed with this, and it turns out that there's just this constant assault of inflammation in the kidneys. There's no repair, it's just more damage. And if there's more damage, more cells will die, that will call more immune cells, that will call more cells to die, that will call more immune cells. So it's a part of a vicious cycle. Now, this is where mine and Emily's favorite protein in our body, I would say. It's called the inflammasome. And the inflammasome is a specific part of the immune system, and it's really the master of inflammation. There are many different types. We'll talk about some in a second. And the inflammasomes are found in immune cells, in macrophages that we're interested in. Now, the inflammasomes are so powerful in inflammation, they require two signals to be activated. So the first one is, I think I should activate some inflammation, but the inflammation that I'm gonna activate is so strong. Are you sure? Are you absolutely sure that you want me to do this? And then the second step is a signal that says, yep, inflammasome activate. And this is usually activated when we get bacterial infections. So it's really great at calling on the immune system, talking to other parts of the immune system to really jump on that bacteria and kill it when we get infected. But we're really interested in a specific type of the inflammasome called NLRP3. Don't worry about what it means, doesn't matter. But basically, this is the inflammasome here. And it's made up of 3 key parts. It's made up of NLRP3. It's made up of ASC and caspase 1. And what happens is when this inflammasome comes together and is activated, we get the release of 2 molecules called interleukin-1 beta and interleukin-18. And these are the molecules that then go on and talk to other parts of the immune system to cause massive amounts of inflammation. Now, the reason we're looking at NLRP3 is because it's the most well-studied inflammasome in kidney disease. It's been looked at in high blood pressure. It's been looked at in ischemia reperfusion, which is a transplant setting. It's been looked at in diabetes, in preclinical models, which are animal models, but the inflammasome has also been shown to be really important in these diseases in humans too. So the question is, well, if the inflammasome is activated in disease, can we stop the inflammasome and therefore we don't have as a significant disease burden? So previously, animals that are missing any parts of this inflammasome, and we can use kind of really sophisticated techniques in the lab to do this, animals that don't have the gene for NLRP3, ASC, procaspase 1, interleukin-1 beta, or interleukin-18, they are all protected from kidney disease. So they have less kidney scarring, less kidney inflammation, they have better kidney function, and they're protected from high blood pressure. So these studies really show us that the inflammasome, which is housed in macrophages, is really important in kidney disease. It seems that when this thing is activated, we get a lot of kidney disease. So, well, then why don't we stop it, like I alluded to before? And there's lots and lots of research going on in this field at the moment. But our favourite drug of choice that we are using to try and stop the inflammasome is called MCC 950. And our lab has actually shown that if we give mice high blood pressure and then we give them this drug, these mice have better kidney function, less kidney inflammation, and less kidney scarring when they take the drug. So, all right, that was a lot from me. So just to summarize what we do know. The immune system's involved in high blood pressure, as Emily showed. Inflammasomes play an important role in kidney disease. The inflammasomes are in macrophages. The immune system, like I said, mainly macrophages, are involved in cyst development, or PKD development. And we know, I'm sure people in the audience could attest to this, people with polycystic kidney disease have cysts on their kidneys and they have a high cardiovascular burden, which is basically just means you have high blood pressure. So we designed a study around knowing these things. Because although the inflammasome has been studied in other kidney diseases, it had never been studied in polycystic kidney disease. So we wanted to ask the question, does the inflammasome play a role in polycystic kidney disease? And if so, can we inhibit the inflammasome to help, reduce the number of cysts and reduce blood pressure in our study? So the way that we did that was that we had 4 groups of rats. So we had one group of healthy rats, we used males and females, and we had one group of rats with polycystic kidney disease, and we treated them with water, basically. Then we had another healthy group of male and female rats and another group of rats with polycystic kidney disease, and we treated them with our drug, with the idea that the drug treatment would reduce those things, like I said, blood pressure, reduce blood pressure, and reduce the number of cysts on the kidneys. This was an intervention study, which means that the animals already had PKD before we started treating them with the drug. And we treated them daily. We took their blood pressure, we collected their pee, we did ultrasounds on their kidneys, and when they were 16 weeks old, we then took all of these different organs to have a look to see whether our drug changed anything, which we were hoping that it did. Emily, over to you.
Okay, so the first thing we wanted to look at was blood pressure. So if you look at this graph here, this is our weekly blood pressure for our male cohort across the entire experiment. So what you can see here in blue is our healthy placebo. And in the pink here, we have our PKD placebo. So as you can see is our PKD rats had much higher blood pressure than our healthy ones, which is what we expected. Next, we have our healthy being treated with the drug. And what you can see is similar to just the healthy receiving the water, they have the same blood pressure levels, which is what we expected, showing that the drug did not negatively impact these animals. Now, what we wanted to see was if our drug could reduce the blood pressure for our PKD animals. And what we found was at 10 weeks old, we noticed that there was a decrease in blood pressure. However, this did not remain, as once we went on in the experiment, by 15 weeks at the end, the blood pressure went back up to normal. So whether there was an immune response at this 10 weeks time, 10-week-old time point, it could have been. However, by the end, our drug did not have an impact on blood pressure for these PKD animals. So next, we decided to also look at our female cohort. Now, similar to our males, we can see that our healthy rats in blue and green are much lower than the diseased rats. However, overall, the females actually had much lower blood pressure compared to our males. But what's different is that in our PKD animals being treated with the drug, there was no difference at that 10-week, but instead at 15 weeks, we noticed there was a drop in blood pressure. So overall, our drug did not reduce blood pressure in these animals. Now, because the blood pressure did not change, we wanted to see whether kidney function was different. So first up, we looked at a urine test. So we looked at the urine taken at the very start of the experiment and the very end of the experiment. And this is a way that we can see how well their kidneys functioning. So once again, in the blue and green, that's our healthy animals, we can see that they are much lower than our PKD animals, meaning that they got significantly more reduced kidney function. So this was then followed up again in another way to test how well the kidney's functioning. And a way we do this is using serum. So like many of you probably have had a blood test to see how well is your kidneys functioning. We took some blood from these animals and then wanted to see how well their kidney was functioning. And what we saw was similar to our urine results. Our diseased animals being treated with our placebo had much worse kidney function compared to our healthy ones. Now, what we expected was that potentially our drug could have helped with the kidney function. So we expected to see that the PKD levels went down. However, what you can see is that they remained the same, meaning that our drug did not improve kidney function. So next, we decided to have a look at the weights of the organs. So the reason we decided to look at the weights of organs is typically if an organ is heavier, it typically means that it has more disease. So first up, we wanted to look at the kidney. So as you can see here, the PKD kidneys are much heavier than our healthy ones. And this is mainly due to the large amount of fluid-filled cysts in the kidney. So as we thought, well, what we thought was going to happen is that hopefully our drug could reduce the number of cysts as well as how big they are. However, if you look at this graph here, the drug did not have an impact with reducing these cysts, so the weights were the same. Next, we had a look at the hearts. So as you can see here, the hearts are much heavier in our PKD animals. Now, the reason behind this could be the fact that these animals have high blood pressure, as you saw before. So high blood pressure can often lead to enlarged hearts and some scarring, which can cause them to become heavier. Now, what we expected was that the hearts may have been reduced, but similar to our kidneys, the drug did not have an effect at reducing the heart weights. And that lastly, we decided to look at the spleens. So as we mentioned before, we are super interested in the immune system, and the spleen is actually an immune organ. So with a increase of weight in the spleen in our PKD animals, it could show that the immune system is actually actively working. So being treated with this drug, which is meant to specifically aim for this, for the immune cells, we expected it to be lower. However, the drug did not seem to have an effect at lowering the weight of the spleen as well. Okay, so next we kind of wanted to have a look at what the kidneys actually looked like. So similar to the images Brooke showed earlier, we decided to look at our own tissue. So what we did was basically cut a bunch of slices and then stain them to see the different structures of the kidneys. So where the arrows are pointed at the moment, we have the glomerulus, which is that filtration part of the kidney, as well as some healthy vessels, as well as some healthy veins, which the arrow is now pointing at. Now, comparing this against our PKD kidneys, we can see that there is a lot less healthy structures, as well as a lot of these blank white areas. Now, these areas are actually cysts that are within the kidneys. So the main thing we wanted to have a look at here is to see whether our drug reduced the number of cysts as well as the size of them. And we did this by counting them. So what we found was in our drug, our drug actually did not reduce the number of cysts. So we wanted to look at the area to see whether they change size instead. However, similar to the number of cysts, the drug did not have an effect on this. So going on, we wanted to see another way, another form of kidney health, which is if the kidney is actually getting scarred or not. So similar to the last images, we cut up the sections and then stain them with a different type of stain. This one instead shows all the healthy tissue in yellow, and then any tissue that has any scarring, it shows up red. So here we are with our healthy ones, and then below we've got our diseased kidneys. And as you can see, there is actually quite a lot of red in our PKD rat tissue. Now, we then had a look at a way, we then wanted to compare how much healthy kidney tissue versus scarring in both our healthy rats and our diseased rats. So in our placebo, you can see that you can see in orange here, our diseased rats have a lot more scarring than our healthy rats. And then being treated with the drug, we expected the scarring to go down. However, it remained the same. So this kind of tells us why the kidney is also lacking some function. Okay, so this is the exciting stuff. Well, pretty exciting for me and Brooke. So as we mentioned before, the inflammasome is very, very important and is probably one of our favorite proteins. I will agree with that. So one thing that we decided to look at was to see whether the genes of these proteins were switched on. So what we have here are the different components. So we've got the NLRP3, we have the ASC, we have the procaspase, as well as some of the products, so interleukin 18 and interleukin 1 beta. Now, we wanted to see if these were up in the diseased animals. So what we saw was in all 5 of these graphs that all of the PKD animals actually had an increased level of these inflammasomes. These inflammasomes, which is very, very cool because this is the first time it's ever been seen in a PKD model. So very exciting stuff for us. So going on from this, we just wanted to see whether our drug, which is meant to target this inflammasome, whether it will decrease. And what we expected was that all these inflammasomes would be decreased after being treated. But unfortunately, what we saw was that there was no difference between our control, our placebo and our drug. So in the NLRP3, in the ASC, in the procaspase-1, and in the interleukin-1 beta, the levels stayed the same. However, there was a bit of a difference in the interleukin-18, but we're not entirely sure what's happening there, so we're going to have a look further on in another study. So why is this happening? Why was the inflammasome we were targeting up? However, the drug that targeted that inflammasome did not work. Well, there could be another inflammasome playing a part of this. So we all know the NLRP3 is one type of inflammasome, but there's actually many different ones. So some other ones are NLRC4 and NLRP6. So these also can work as a way to prevent, as Brooke was mentioning before, how NLRP3 attacks the nasties, similar to these. So we decided to have a look to see whether some other inflammasomes were activated. And what we saw was NLRP4 and was upregulated by a lot in these diseased animals compared to our healthy ones. And then looking at NLRP6, we noticed that our diseased animals was actually much more downregulated than our healthy animals. So this is very interesting. So we looked at it when we treated it with the drug, and surprisingly, there wasn't any difference. The reason being is our drug was meant to target NLRP3 specifically and not necessarily these two inflammasomes. But this is very, very interesting because maybe these are actually playing a role in PKD, which is why the drug isn't working.
Okay, so Emily just took you through what was happening with the genes in the kidney, but what actually is happening in the kidney? So we used some imaging techniques to see if we could look for some of those proteins of the inflammasome and where they were on the kidney. So you can see in our healthy kidney, we don't really see any staining. What we were looking for was macrophages, which are in green, and NLRP3, which is in red, and DAPI is just basically a marker that tells you where a cell is. So in our healthy kidney tissue, we didn't see any macrophages and we didn't see any NLRP3, which is what we expected. However, if we have a look at our PKD tissue, you can see all of these green dots here. They are all indicating macrophages. So we've found macrophages in our kidneys, same as on this section here. And if we take a really close look at one of the cells, you can see that it's got a bit of green and a bit of red. So this is a macrophage that actually has the NLRP3 inflammasome in it, which is in our PKD rats. So it was also really exciting to show that this stuff is actually in the kidney as well. Then we also did some staining for ASC, so another protein of the inflammasome. And you can see here in our PKD kidney, this is not treated with our drug, but just a diseased kidney, an untreated kidney. You can see all of these big cysts here, but you can also see on a couple of spots these red dots and over here. So these red dots indicate to us that ASC and the inflammasome is in this kidney too, which is also really exciting and no one's ever shown before. But as you've probably guessed, this is animal tissue. So this comes from our rats. What we also wanted to see is if the inflammasome was activated in human kidney cells. And when we grew human kidney cells in low oxygen, remember that was a stress that caused kidney disease, we can see these red spots start to appear in these cells. And these red spots are also ASC. So again, this tells us that ASC, and therefore part of the inflammasome, is activated in our PKD kidney, in rats, also in human cells, which is really, really exciting. But what does this all mean?
Okay, so what we have found so far is that our PKD rats have higher blood pressure, they have worse kidney function, they have large cysts on their kidneys, but they also have heavier kidneys, hearts, and spleens compared to our healthy rats. We also found that the NLRP3 inflammasome and its components, as seen in the diagram again, is increased in our PKD rats compared to our healthy rats. However, when treated with the drug, it did not reduce blood pressure, cyst growth, or scarring, and it did not improve our kidney function. However, we did find that it significantly lowered our blood pressure in our male rat cohort at 10 weeks. So what is next? So we have a couple of questions left to answer. The first one is, what is happening at 10 weeks? Is there a difference in the immune response that's causing that increase in blood pressure? And also, are there any other parts of the immune system, such as NLRP3 or NLRC4 or NLRP6, playing a role in cyst growth or high blood pressure? And the way we're going to do this is just do some further studies looking into this sort of thing. Beautiful. And I'd just like to acknowledge everyone who's helped us with this project, but also a very special thank you to PKD Australia and Australia Rotary Health, who have given us funding to work on this project.
Thanks, Jess. That's all we've got.
Thank you.
Thank you. Thank you so much. Okay, well, before we launch into the questions from our attendees, I actually wanted to ask you both how it is that you ended up doing research for PKD and that, connected to PKD. So, Brooke, you want to start with you?
Yeah, sure. So I have kidney disease myself. And from a really young age, I was around doctors and things like that. And I was always interested and really good at science when I was in high school. So understanding how the world works through science became a real passion of mine from a young age. And then I was working a full-time job and I had a kidney transplant, 14 and a half years ago now. And so that was time where I kind of reassessed my life and what I wanted to do. And I started to move into research. And for me, researching kind of my own kidney disease is a bit too close to home. So I'm really interested in the genetics and everything and with PKD. And so, that started to be my kind of area of interest.
Wow. Okay. Thank you. Just while we're chatting, feel free to put questions in the Q&A if you have any. And then Emily, how did you get to researching about PKD?
Okay, so similar to Brooke, as a kid, I was super interested in science, super interested in helping people, but I didn't know what I wanted to do really. So I did my undergrad, and then once I finished my bachelor's degree, I realized that I really, really liked working out how things worked. I liked working out how like if something goes wrong, how we can fix it. And most importantly, I knew I didn't want to work with tiny little cells as such, and I would rather work out how a whole system works, which is why I kind of stumbled into kidney disease. Luckily for me, I soon realized that I do also like how little things work. So lucky for me, I get the best of both worlds with this project.
That's so interesting. Thank you. I love to connect how our researchers connect with PKD, particularly for those members in the PKD community that we have here today. All right. Well, no questions have come through yet, but I did send you quite a few that were sent through upon registration. So if there were a couple there that you wanted to answer.
Yes, the first thing that I wanted to say to everyone, which is that Emily and I are discovery scientists. That's, I guess, how we're differentiated from clinical researchers, and clinical researchers are different from nephrologists as well. Although a lot of nephrologists also do clinical research. So a lot of the questions that were about kind of nephrology, or talking to a kidney specialist, Emily and I are not experts in that, so but they're super interesting questions, and I think nephrologists, my experience with them in both research and kind of personally, has been that they love having these discussions with their patients. So anything that you don't feel like we answered or that we can't answer, fully recommend you go and talk to your nephrologist. A good thing could be, hey, I went to this webinar and I had these questions. As a good starting point. So yeah, if we don't if we don't answer a question, definitely talk to your specialist about it. I guess, Emily, there are two here which I think are more related to our research, which is, what are you most looking forward to in the future, and what has you found most difficult?
Okay, well, I'll start that off with what I found most difficult. So as just kind of explained, this project has been a long time. So we've been working on this since 2022 and we still haven't quite finished it. So what I found the most difficult is sometimes science works and sometimes science doesn't. So what I found most difficult is just trying to get things to work, which is probably why it's taken so long, but we're getting there and hopefully soon it'll be all finished.
And what are you most looking forward to in the future?
Ooh, most looking forward to answering the questions that we have left over. So what is actually causing it? Whether it's the other inflammasomes is super interesting, but I think what I'm most interested for is what's happening in that blood pressure. So I don't know, super interested in that part.
Yeah, I think for me, yes, I agree, Emily. Science always throws little curveballs at you, which makes it a little bit harder. Not harder, it throws curveballs at you that keep science interesting, but does have some setbacks. So those setbacks are always challenging. And I think a lot of the science that we do is about figuring out what doesn't work and then what does work. So while it might seem like a lot of our study was negative, as Emily said, there's some really interesting things to follow up on, which I'm really excited about. So this kind of experiment gave us different leads to go down different research arms, which is really cool and exciting. Jess, I can see some things coming up in the chat. Did you want to?
Yes. So one question has come through asking whether the next part of the study is to look at the other two NRLS and the drug, and if so, what would you expect?
Yeah, that's a really good question. And in fact, when we first started this project, the drug that we used, MCC 950, had really just been developed. It was new. It wasn't on the market. It was for research only. And we could only get it from a collaborator who was in Queensland. So, and she's a chemist, and all she does is kind of create new drugs. So she's a really good person to have around. But the kind of problem is that not a lot of inhibitors are available for the other NLRPs. So yeah, we would love to follow this up with other inhibitors of that inflammasome, of those different inflammasomes Emily mentioned. But there aren't any kind of drugs available to do that yet. We'd need to work with, I guess, chemists who are developing these drugs to see if it's something that they could help us develop. Anything you want to add, Emily?
Not really. The only thing is, like, if something like this does come up on the market, even though our drug didn't work, we may have seen that other drugs could be also influencing it. So maybe even if a different drug doesn't work, maybe multiple could work together. So it's always a world of possibilities when it comes to finding new drugs and seeing what works.
Thanks. Okay, we've got another question just come through on the Q&A. Will you be looking further at the drop that happens in males at 10 weeks?
Yeah, yeah, absolutely. I think that's what that was so unexpected for us. We did not expect to see the drop and then it come back so early. So we think that maybe something is happening with the immune system, like Emily said, at those 10 weeks. The drug is doing something, we just don't know what it is. So we are absolutely doing another experiment now, actually. We're trying to understand what happens with the inflammasome over time. So in this study, we looked at one time, which was when they were 16 weeks old. But now we're going back and trying to find out what happens in between those 16 weeks so that we can understand what might be causing that blood pressure drop at 10 weeks, because if we can understand that, then maybe we can do something to help that drop stay on, help that lower blood pressure continue. So
Wow, well, it sounds like we might need to have a part 2 to this webinar.
maybe in another 4 years, 2 years, 4 years.
Yeah, give us a
It
couple
takes
years.
a long time. Discovery research does take a long time. So yeah. There was another question on polycystic liver disease, which I found really interesting. So in the model that we use, they sometimes do get polycystic liver disease. So while I don't know specifically about the liver, I would assume that a lot of kind of chronic conditions, like things that affect the liver and things that affect the kidney, are made worse by the immune system. So I would suspect that there's something similar in relation to inflammation going on in polycystic liver. There was also a lot of questions, which I'm super excited about, but I don't think I have a really good answer for anyone, about the gut and what we eat and how can we maybe change our diet to have a low inflammation diet. And I can tell you that there is a lot of research going on with how the gut works and how the gut itself might contribute to hypertension. So, having a healthy diet, and healthy diet just means lots of fruit and veg. There was someone in here saying something about not eating meat. I don't know about eating meat or taking supplements, but yeah, generally a healthy diet is a good way to kind of keep inflammation at bay. But other than that, I can't tell you exactly what, because it's still a heavy area of research.
Yeah, there were a lot of questions that came through about that, actually. And as you say, there's a lot of research currently happening. Do you know if any have progressed to clinical trials? Because there was one specific question about whether there were any trials planned in relation to that connection between diet, gut, and inflammation.
So I don't know of any specifically. It definitely doesn't mean that there aren't any out there. There are, I don't know if people have heard, or maybe Jess, she might have spoke. If not, maybe she might be a good person to get to speak, but a researcher, Professor Kelly Lambert in Wollongong. Yeah.
Yes, she's been on a webinar, and earlier this year, we shared some of her recipes and her diet and PKD, and
yeah, she's massive
about that
in this field, and she's an amazing researcher and everything. So yeah, there's definitely, like I said, she's doing a lot of research as well, so there's definitely a lot out there.
Excellent. She's actually just joined our scientific advisory board, so perhaps I need to have a conversation with her, since there seems to be so much interest about how diet is affecting PKD.
I mean, I just want to say that's amazing that the patient group are so invested in this. Like, I love to see that. It's great that people are thinking about how to slow their disease or to take care of themselves and things like that's just so, I think, amazing to see. Yeah, so everyone keep up the good work.
Yes. Okay, one more question before we finish up. Just a question about your discovery research, and can you answer historically how has discovery research led to changes in treatments of the CKD, PKD?
Yeah, so like I said, it takes a long time. But I think the biggest good news story that's come out really recently is, I don't know if anyone's heard of SGLT2 inhibitors. It's not, I don't think it's kind of used for PKD, but it's definitely used for diabetes. And SGLT2 is a receptor that's in a specific part of the kidney that was discovered like 80 years ago. And it, based on that discovery, eventually we've realized that if you inhibit, it's a receptor, sorry, like I said, and if you inhibit that receptor, you can kind of really change the lives of people with diabetes. They don't have as high glucose, which means that in their blood, which means it's not damaging end organs like the kidney. And so I guess that's the best kind of like, that's what everyone wants to do, right? Find that thing that then leads to like a really good outcome. And so they've actually started to use these SGLT2 inhibitors for just general patients with chronic kidney disease. Helps a lot with weight management in people that might have hypertension, induced kidney injury, helps reduce their weight and things. So they've really, I think, changed the game for a lot of people. And it all started because a discovery scientist discovered the receptor. So
Amazing. Well, on behalf of everybody who attended tonight, our wider PKD community and our board, thank you so much for sharing your knowledge and just all the hard work that you have been putting in and will continue to be doing in research in this space. Thank you for sharing about, I learned so much tonight, everything from microscopic cilia to bonus immune system of the axolotl, and then your favorite protein, the inflammasomes. Yeah, I've learned so much and we're really, really grateful. And thank you for everyone for coming as well. And stay tuned for our next webinar. This will also be on our YouTube channel shortly. So thank you and have a great night.
thanks, Jess.
Thank you.
Bye.